Hi, if you've reached this point, you've decided to take the automated tour of the NADH dehydrogenase complex as part of the 3D models set up within this Minecraft world. So if you look at the position there on the top left, 290, 87, minus 52, that's where you'll find this door entry bot as we were, which is also part of the story mode. You can read the blurb there, again using the immersive reader if you like to assist you, but the basic idea here is to click the entry point. Now this will take you to an area which is now elevated. You were down the bottom there a little moment ago, but you'll have some options for free play and roaming as well, and you can do this later, but if you wanted to take the automated tour, try not to set off some of these plates, which is going to bring up a bunch of dialogue, which I'll show you right at the end. You want to interact with this NPC. Read the blurb here. I presume you've accessed the support video either using this NPC by clicking on that link below, or you've accessed this using the website version of this directly. But bottom line is, if you have this video now, you want to start taking the tour. We're going to time you in when it's appropriate to click that Start Tour button to make sure that the narration is aligned with your teleportation. So don't click Start Tour until we count you in. So get ready to activate that Start Tour button using whatever controls you need. I'm going to count you in. You have to, when I say go, you're going to have to hit that button, not just then, but I'm going to count you down from 5. And then get ready to click that Start Tour button and we'll go to our first location. So you ready? We're going to say 5, 4, 3, 2, 1, go. Start tour. So this has teleported us from that platform just to the other side. And now we're facing a large display which is meant to mimic some of the respiration aspects of the cell and energy production system. So we've got glycolysis written up there and the TCA cycle, also known as the Krebs cycle, funneling energy in the form of electrons to NADH. And these you can see light up arrows which then proceed down through this complex. And while we're not trying to go over earlier content that you might have seen in first year biology on glycolysis and the TCA cycle, we do want to reflect on the fact that these electrons supplied by the breakdown of foodstuffs flow through electron carriers such as NADH. As the arrows light up here too, you'll see them in sequence light up a series of lamps that come down through this complex. The arrows show you the direction of flow of those electrons, and you can see over here the 4Fe4S. This is meant to reflect the first iron sulfur cluster, and then the second one, the third, and also coenzyme Q right at the bottom. Now, what's just happened then as the jolt occurred is that teleportation has moved us over so we could activate another plate. And by the way, don't move too much in terms of forward and backwards off these plates or you'll wreck the sequence, but you can turn left and right. But what's now happened is we've mimicked the effect of having no oxygen. So now electrons no longer flow down through these complexes. They are diverted from NADH over to fermentation, which just reminds us again that the cell has different ways of dealing with these electrons if oxygen is not available. But you'll now notice that the electron flow down through this complex has now ceased. And we're going to take an opportunity to look at that more carefully. But before we leave this location, just notice the CoQ at the bottom there. This display shows you the flow of electrons all the way from NADH down to coenzyme Q, also called ubiquinone. And each one of these little blue structures reflects the different types of iron sulfur clusters found within complex 1. So we're going to spend a little bit of time teleporting around complex 1. In a moment we'll re-emerge on the other side where we'll take a closer look. And if we turn to the right, this large green rectangle in the middle with the two little lamps which are yet to be lit up reflect NADH. Now what's happened now is we've jumped off that teleportation plate which also turned on the anaerobic conditions. So now oxygen has become available again and the electron flow will now continue down through complex 1. And in some cases we won't be able to see everything, but as we teleport around you'll get better vantage points. You can see down here some structures which has some iron molecules and some sulfur, and they're meant to reflect the iron sulfur clusters. The yellow are meant to reflect cysteine residues within a protein that holds the iron sulfur cluster. At the back there is actually a repeat of this front structure, so it's been replicated except we've coated that NADH dehydrogenase complex structure with blue blocks, reflecting the fact that these electron carriers are within a complex. So we'll gradually teleport around this initial structure and get to the one at the back there. So if we turn back around though, now we can see the electron flow coming through the back end of NADH dehydrogenase, and I've increased sort of the guardrail here so you can't move around, so you can't see a huge amount, but as those electrons flow down you can see some boxes there with some carbons and oxygen, and that is meant to reflect the flavin mononucleotide structure, which I didn't mention when we were standing at the front, but FMN, you would have seen it from the other side, reflects the flavin mononucleotide that's one of the electron carriers within NADH, dehydrogenase. And in fact, it's the first electron carrier that captures the electrons one at a time from NADH. So they're passed on. And if you go down and explore that structure directly, you'll see that the lamps for NADH dehydrogenase light up, and then two lamps will light up for the FMN, reflecting the addition of one electron, where we get the flavin, the flavin semi. Got to remember what this is. Um, flavin semiquinone. See, my brain's pausing there, can't multitask. And the addition of the second one will have the fully reduced form of that carrier. So the electrons flow through the flavin mononucleotide, reducing it, and then those electrons will subsequently be passed on to the iron sulfur clusters. Now, of course, what happens then is the flavin mononucleotide carrier itself becomes oxidized again and can take up the next series of electrons. So you can see now these lamps lighting up within the iron sulfur clusters happens in sequence as those electrons flow and ultimately get to the bottom of this structure, which is where we've added in some extra carbons, nitrogens and oxygens to reflect the structure of ubiquinone, which of course can also carry two of those electrons. So you'll see a couple of lamps associated with those electrons arriving at the ubiquinone. And we'll teleport down closer to those locations so you can see better. We'll do so on this blue structure. So this is now essentially the repeat of that structure at the front, where if we see in reverse, those electrons flow down from NADH into the flavin mononucleotide and down into the iron sulfur clusters. And that's happening again here, except now we've encased this version of the complex with these blue blocks, just making it feel a little bit more authentic. But here we can actually see those two lamps, if I put my crosshairs on it, of the flavin mononucleotide picking up those two electrons, forming the semiquinone and the fully reduced version, the quinol. And then those electrons will pass down through the iron sulfur clusters and will teleport down through the core of this protein in a moment. We're almost at the end of this teleportation sequence. We'll end up down the bottom where that blue structure, so the red and blue structure is, which reflects where the protons are being pumped. But while we're waiting for the next teleportation sequence, you'll hopefully reflect on the fact that these electron carriers lie in close proximity, or the correct proximity to each other, which is reflected better here. We can see the first of the iron sulfur clusters here, and you can see the iron and sulfur atoms. The orange blocks reflect bonds, and these yellow blocks reflect cysteines. And you can see that the electrons arrive and come through the core of those in sequence, but they are appropriately spaced so that electron flow does not skip any of these. So electrons must follow a specific path through the core of these proteins. You can actually see at the back of this iron sulfur cluster there's a green block. And this reflects a slightly different type of iron sulfur cluster called a Riesky, or a Riesk if you would prefer to say it that way, iron sulfur cluster where there's a histidine taking the place of what would otherwise normally have been a cysteine. Now this makes, of course, the iron sulfur cluster behave a little bit differently because it's the iron within these molecules that hold transiently those electrons between them. And of course, by changing the local environment around these iron sulfur clusters, including the addition of the histidine, you can therefore change the reduction potential. And we're not going into reduction potentials much on this display, but you can certainly learn more about those in the other parts of this module. So you can see now coming from the bottom of this display the electrons flowing down through the iron sulfur clusters in sequence, being passed from one internal carrier to another. And here's our two lamps reflecting the electrons being added to ubiquinone. So it becomes ub semiquinone and then ubiquinol. So that one lights up first in the background and this one closer. And this structure is meant to reflect the spatial positioning of the atoms within ubiquinone. You can come down here and explore as part of free play using the follow along videos where you can jump on different plates and look at the effects of adding inhibitors such as rotenone to the electron flow, where you would look up here into the structure. But for the rest of this teleportation sequence, we're going to spend our time over at the proton pumping part of this display, which we'll teleport to in just a moment's time. But you can see over there in the distance the hydrogen blocks coming out. They're meant to reflect protons or hydrogen ions, and this complex pumps four of them. And so behind this structure would of course be the matrix, because the pumping occurs from the matrix to the intermembrane space, also often referred to as the intercrystal space. And you notice how these little lamps lit up? That is just meant to reflect the electron flow coming down through this complex and the timing of proton pumping with electron flow through the complex. So some of the energy harnessed from the electron flow through complex 1 is used to generate this hydrogen ion gradient across the inner mitochondrial membrane. So of course these structures would normally be within the inner mitochondrial membrane. In the background you can see some unfinished work on succinate dehydrogenase and some other complexes, but right at the very end of this display over, if I was to go in the distance in this direction, you notice where your position is, 364, 92, minus 46, and we're going to teleport down in a moment. You can pick up an interchange to take you down to a rather more completed version of complex 4. So we've got basically on our left here complex 1, which picks up electrons at one end of the electron transport chain, and complex 4 right at the very other end. But we've got some unfinished structures here from succinate dehydrogenase, coenzyme Q is part of both the NADH dehydrogenase and the succinate dehydrogenase structures. But then the electrons will move on to complex 3 and then cytochrome C, and they're the ones that need a little bit of work. But if you want to take the tour again and do some free play, you would essentially just click on the retake tour button. But if you think you've had enough of this display and just want to teleport out onto the other side of these walls to catch a ride somewhere else, just click on the teleport button. So I'll just retake the tour for a moment just to show you that if you do come up here as part of free play, you can jump on these different plates and activate a series of text whispers, as it were, or dialogue that you can read. If you don't read everything, just jump off and hop back on again if you've the timing was a little bit too quick for you, but you notice there are several prompts that come up. So please make sure that each prompt has finished before hopping off the plate. It should disappear. And then when that occurs, you can move on to the next plate. In many cases, I've been mindful enough to write the end, but sometimes I believe I've missed that. So that sort of commentary starts by reading text 1. You can hop onto text 2 and so forth, text 3 and 4. You can also come along here and activate that anaerobic situation again. And if we do that right now from this position, you can see that's meant to reflect that signal going over saying oxygen is no longer available, and this will trigger a change in the way that structure functions. So the electron flow now no longer goes through that structure in the distance. We'll wait for that sequence to finish there, and now those electrons are bypassing from going from glycolysis into NADH over to fermentation rather than through the electron transport chain. Of course, you can see there's some dialogue popping up as well at the same time. So feel free to explore this structure. When you're finished, you can just go along the gangway, as it were, and jump on a series of other plates to get some little bit more information, etc., and some commentary. And you can interact with the story mode 3D models bot number 3. So you pick up the third video, but where to find each of those videos for the story mode is a separate entity and appears both on the YouTube channel as well as our Blackboard learning platform system in the correct sequence. So you'll be able to find them there, and they will take you to where you'll need to be to pick up the next part of that display. And you can see in the background there the complex 4 part of this display that I was referring to, but you'll have to catch a cart or fly down there, all the way down there, to pick up that sequence. So thanks very much, and I hope you enjoyed that teleportation sequence. Bye.